Just about everything inspired me, but obviously talking about the individualized cancer treatment, I think is the most critical for me personally. We actually went down that road when Audra had her cancer and we were exploring it. And I've worked in the space of CAR Ts and cell therapies that are really more one-to-one versus one drug hits all approaches. There are some that are great, like some of the PD-1 cancer immunotherapies have had a positive impact. But I think at the end of the day, this idea of individual sequencing that we can do ourselves, that can help you engineer, if you get any disease really, but engineer a treatment to attack that disease. I mean, that seems to be the future. And the big issue is it's scalable? It sounds like we're not far from that. And the reason I believe it is the taking off of the GLP-1 space. And now I think we're going to see it in hormones. People are taking more control and responsibility for their own health and realizing, okay, I've got to sequence and, I've got to know what is happening. And I do think we're going to move in a direction that is very, not self-diagnosis, but I would say better informed health planning for your whole life. That's been our theme here. And I think it was nice to hear reassurance that those technologies could become reality, because you always hear how they're not cost effective and they'll never be scalable and it'll take billions to do it. Well, what better way to spend billions? I think people should listen to this episode mainly, I think, to reduce some of their fears in and around the use of AI in medicine and in healthcare and tech in general, right? Because I think tech was always interested in the health and science areas, but it's really going to accelerate things and expand our ability to do more versus contract that. We have technologies that can help us overcome a lot of obstacles, and I think it's just the half glass full version. A lot of people are pessimistic about what the world will be like for our kids and their kids, my grandkids, and all that. And it's like, oh, it sounds like it's going to be really freaking cool, obviously, if it's applied the right way. Hey, Andrew, thanks for coming on.
Hey, it's my pleasure, man.
How did you get into this line of work? Has it gone as you expected?
Oh, no, I've had no plans. There's been very few expectations. I was just someone who was always interested in how things work. Always. Like, I remember distinctly asking my parents for those books at Christmas time, how things work. And I'd just go through them cover to cover. Most machines back then were pretty simple. I was born in the 1960s. And in my teen years, as I'm going through high school, I was working with everything mechanical, electrical, starting to get into the first PCs. But I realized the one thing that nobody really knows how it works is biology. And that's where I decided I'd focus. And when I switched to essentially Bio 101 and the whole program in university going through life sciences, most of my peers were people that were just going into so they could go into medicine because you needed a couple years of introductory biology. Whereas I was completely focused on single-celled organisms, the basic machinery of biology of the cell, and wanted to know how the cell works from a fundamental level. So that's shaped most of my career, as well as drawing analogies to the electronic world, to the computational world, because it turns out that cells and computers, very different mediums, but they share a lot of the same architectures.
And the worlds are colliding. I mean, I remember I started my biotech career at Genentech in 1992, and Art Levinson was the head of R&D. And, I remember him being very focused on computers and technology and data in ways that, I hadn't really appreciated fully, and how they put all that together to design, really great clinical trials. And obviously, they became incredibly successful. You basically said the age of programming life has arrived. What does that entail? Like, it sounds a little scary, but maybe not. You want to talk about that?
Yeah, I'm happy to. So our career paths are probably similar in some ways because I joined Amgen, Genentech's probably main competitor, in 1995. And I came in because I recognized that we were entering a new age in genomics. The Human Genome Project was 5 years in. I had seen new types of sequencers on the bench. They hadn't even been, it was still several years before commercialization that were game changing. I recognized that I really needed, it was time to move into life science. And I was brought in because I was one of the first people kind of working in bioinformatics. I was using computers every day to analyze genetic code, build databases, process information. That just wasn't a common skill in the folks in life science at the time. So it was a really good fit for me. That being said, going back to programming life, I saw very clearly from the earliest days that the cell was a processor. It wasn't like a computer processor in that a computer processor processes bits of information. This was processing molecular molecules. But if you drill down, it had an operating system that was written in DNA. And of course, the whole Human Genome Project was to understand our operating system. But because I came from the world of microbiology, I was super interested in understanding the microbial operating system. As the Genome Project was going on, we were getting more and more data. We were starting to see how the code was put together. And after the Genome Project was completed and a number of bacteria had been sequenced, it was time to move away from reading the genome and start thinking about, how do I program it? How do I start writing?
Is there a biodictionary? I mean, can we separate some of these areas? There's bioeconomy and biosecurity and biodefense and biotechnology. How should we be thinking about it? With AI, which we're obviously going to get into, in this age of AI, how would you define where we are now?
Because I've always worked at the intersection of computers and biology, for me, it's more of an evolution, but it's still very early days. For example, in 2024, the Nobel Prizes were awarded to researchers that were using AI tools for interpreting the structure of proteins. That was Demis Hassabis and John Jumper at Google DeepMind. And David Baker got the other half of that Nobel for designing proteins. But proteins are the smallest components in cells. You can think of as basically the biological equivalent of the transistors or other components in a computer. And so we're really going bottom up, but those tools are now intelligent. Just in the last 5 or 6 months, we've seen the first AI designed viral genomes. And we can build AI tools to design, for example, a bacterium, but we can't easily build and test that yet. So we've kind of got more computational power on the design side than we have automation and technology to do the compile and boot up, if that makes any sense. But these two fields are completely, AI is going to take over biology and allow us just to start to program biology by clearly defining our intentions. Think of it as vibe coding biology.
Should everyone listening to this be freaked out by that? Or how do you think about it? Because some people might say, oh, you know, the machines are going to create, that's where this is going to go the wrong direction. But I guess we're probably similar minded, you know, in that, no, it'll facilitate, it'll help us maybe get to some real big answers faster. But I'm not that fearful of it, of the unknown. But how would you help people understand it, what the role will be?
Every technology has a good side and a bad side. Every technology is dual use. You know, a fork can pick up your food, it can poke out an eye. So the technology itself is agnostic. It's how we use it. Most people, well, you know, 99.9% of people are going to use it in a positive way or in a way that is economically positive. Let's put it that way. They're going to try and do something productive that earns them money, that helps their family, that solves a problem. They're going to use it in a productive way. And there will always be people that will use it maliciously. They'll find some weak point, they'll scam somebody, they'll target something, or they want to build a weapon specifically, not for defense, but for offense. You can't blame the technology in that. You can only look at the people and how they're using it. There is the potential for accidents. Some people might be trying to do something good and go, oops. Some of those oops can be quite dangerous. My big fear in biology, personally, and people have different ones, is that someone will use these tools maliciously to create a pandemic class virus.
I guess the real question around that is, for all the advances that have been made, why aren't we closer to cures or answers in some areas? You'd think this combination of computers plus biology would have us at the point where things would go fast, we'd have answers. Why don't we have them all yet?
I think we just need to be a little patient. First of all, we started genomics kind of by saying, oh, let's go sequence the human genome, which is the most complex genome on the planet when you think about it. And so we kind of started at the top. And now we're working down and applying the knowledge that we got from sequencing and continuing to analyze human biology is very slow to translate to humanity because we have rules and regulations about what we can do to people and what companies can do to people. So that's why cures are slow. But when it comes to the technology for being able to program biology, that is growing exponentially. Again, the stuff that Amgen or Genentech was doing 30 years ago is available to grade school kids today. And there it might think of it as toy biology or playful biology or lab-based biology, but these tools are extremely powerful. Remember, it takes 10 years to get a new drug through the drug development process. It's extremely expensive, and that limits the number of things you can do. But we are seeing drugs that have been computationally designed going through that process. And just in the last couple of weeks, one of the companies that I've been tracking for years that does design of drugs using these AI-based computational systems called In Silico Medicine just went public in Hong Kong and was hugely oversubscribed, and is one of the first generation of new drug companies that's kind of AI first. None of the other drug companies are AI first, but that's changing.
And why not? I guess they didn't adopt it, or are they going to have to catch up to it? Because some folks have said to me, who run these companies, we don't use AI at all purposefully.
The entire AI can work at every single facet of drug development. This takes me back to about a year ago. I had a contract with a major pharma talking about how AI could be used in their drug development process in clinical trials. And the short version of that presentation and the research I had done for it was it basically sped up the report generation. They really weren't using it effectively in any other aspect of clinical trials. AI can be used at the very start of the drug development process and supported with every step of the clinical trial and research process. So AI first companies that are starting to emerge will have a huge advantage over the classical drug development paradigm. And I'm starting to see that in cancer. We can go into that if you like, but there's a reason for that.
Yeah, I do want to go into it. Yes, I'm certainly invested in those areas and it matters to me personally. What are you seeing there on the cancer front?
So my argument with cancer has always been, we've been going about it all wrong. And that's because no two cancers are identical. 30 years ago, we called it breast cancer, prostate cancer. That's just calling it by tissue of origin, which is determined by pathologists looking at the cell types. Now that we can sequence cells and really inspect their code and how their code is operating, what we call expression, we can really deeply molecularly analyze a cancer. And what the first thing you find out is no two cancers are the same. Meanwhile, the drug development process is geared to making one-size-fits-all medicine. It's a complete mismatch between the problem and the tools we have to solve it. And that's why cancer hasn't really been cured. You can't do a one-size-fits-all cure for something that is individualized. But the most exciting thing I've seen in the last few years is there's been groups now that are taking the entire drug development process for cancer, starting with diagnostics of the cancer cell and making it individualized. starting with one person's cancer, deeply analyzing it, and then using that to develop custom therapeutics and vaccines. So both passive, training your body to recognize the cancer, as well as actively targeting and eliminating the cancer and building that entire pipeline for the single individual. And in fact, it's becoming cost effective to do that because some of the most expensive modern medicines, one size fits all, or slightly personalized around key targets in cancer, are getting up to the multi-millions of dollars. But this entire individualized process is already at about the same price, and it gets cheaper because most of these processes are digital. Everything digital gets faster, better, cheaper. The main accelerant is that now you're not doing phase clinical trials to reduce risk. You could basically have approval to work with a particular patient before you start.
So cells are living computers and DNA is a programming language. Will everything, it sounds like we're going in this direction in healthcare, become engineerable? Can we engineer it all?
Yeah, I think we start knocking off diseases faster and faster. Again, cancer, I think, will become a managed disease. You can't ever eliminate because cells are going to corrupt and start to all the time. I think infections with bacteria, again, people have worried about antibiotic resistance. I think we're in the golden age of antibiotics. We don't need to just use a few chemicals anymore. We have so many new programmable moieties to chase after a particular bacterium. They're clonal. So much easier to kill than a cancer cell, which will have a spectrum of degradation inside a particular individual. We're also starting to see this N of one approach emerge with rare diseases in children, where there's just been no treatment at all. But now through advanced diagnostic technologies, we can get a deep understanding of a particular problem pretty much at birth. This is really fun. And it just keeps going on from there. Our genomes are 3 billion bits of code. How long will it take us to be able to write eukaryotic genomes like our own? At the outside, I think it'll take 20 years. I think it could happen much faster than that.
I mean, you have till 80, bro. So you got to get there. You got to get going. If we can engineer DNA to rewrite the book of life, should we be doing this?
I think absolutely yes. I've never seen prohibition work in any area, from alcohol to nuclear technologies. I think we just have to use it wisely and productively. Life is the most powerful technology we have. Growing something is magical manufacturing, right? It self-assembles. And if you can do it just by writing the program, wow, that's huge. So I believe that biotechnologies combined with AI technologies is a superpower. And we're just learning how to unlock that. And yes, there are real risks, but there are huge opportunities in every aspect of our lives and for taking better care of our planet. Because we've been extremely successful as a species, but we've been using pretty crude technologies to generate that success. And we know that they're not sustainable. It's changing our atmosphere. We're using too much of this and too much of that. We collect all these waste products that can't be broken down yet. Yeah, by programming biology can fix all that and take us to a cleaner, sustainable, infinitely recyclable world. And I think if you can get there in your thinking, we can go and create it.
So you co-founded it way back in 2016, Genome Project Write, GP Write, a community that is writing genomes from scratch. Is that still going? Is that still rolling? And what the fuck is it?
Exactly. So remember, I'm old. So I remember when the Human Genome Project was launched, right? When the Human Genome Project was launched in 1990, it had taken 6 years of planning, convincing Congress to release the money. And, Jim Watson was at the head of it. It was a big deal. And that was just to read our genome. The thing that I recall most about that launch, working in bacterial genomics, was we hadn't sequenced a bacterium yet, the smallest cellular life. We tend to be very human-centric. So after the Human Genome Project was completed, I expected us to take that energy. It was the biggest science project the world had ever done, scientists had ever done. It was kind of the Large Hadron Collider of biology. I expected that energy to stay cohesive and move from reading to writing. Let's start programming biology now. That didn't happen. What ended up happening was the field split into omics, proteomics, metabolomics, glycomics, like 1,000 omics blossomed on that foundation of reading the human genome. And by 2010, 7 years after the close of the project, there was still no effort to talk about let's build a foundation for synthesis and programming, even though the field of synthetic biology was kind of bubbling away under the radar. And so I wrote an essay for the Huffington Post at the time saying, asking the question, is it time for another genome project? This time around writing. At least that's locked into the history books, but it really didn't catch on. And by 2015, I was getting so frustrated. I proposed that we create a new human genome project, the Human Genome Project Right. That got us some traction, some funding to launch, and it went out the door in 2016 with a bang. It was the top 100 paper of 2016. And we had a lot of energy around that. What we didn't have is a big funder that would come in and write $1 billion check. And to my chagrin, the board decided to drop the human part of the Human Genome Project, right? Which totally made media attention nosedive and made it impossible to find a big funder. So short version is since COVID, where we went virtual, since then, we've kind of parked it. I'm the chairman. I maintain everything. It's all there. It's all parked. And what's emerging now with AI, with increased ability to program, with better synthesis and assembly tools, and a UK effort that has emerged called SynHG, Synthetic Human Genome, to build a human chromosome. It's time to reactivate that.
Genome Project Right aims to engineer cancer resistance, viral immunity, transplantable organs. Which breakthrough do you think is closest to reality that you could see come through? Is it the cancer one?
We don't need to write a whole human genome to cure cancer. You just need to build essentially bioweapons for cancer cells. And that's typically going to be small things that are going to be bioavailable. I think the thing that is really going to cause the Human Genome Project Right to take off is building human cell cultures that are resistant to infection. Because we use human cell cultures a lot to make products or do research, but it's really hard to grow human cells in a dish and stop them from being infected from just the viruses that circulate around us. But for me, it's the compiling technology, the synthesis and assembly. Just as we start to put more effort into writing a human genome, the synthesis and assembly technologies will improve, and that will make it easier to start programming bacteria, starting to program single-celled organisms like yeast, et cetera, et cetera. So I think just the core technology is really the most exciting short-term thing to come out of this, as well as the AI design tools.
Are you talking to Kennedy and Dr. Oz and some of these guys about this yet? Or have you been asked to? Will you?
Oh, it's definitely reaching them. I haven't gone to Health and Human Services. That should be fairly obvious. But yeah, I go and try and message this to as much as I can and get the word out to people so that they can start figuring it out for themselves and putting the resources into these technologies. I'd love to see the US have the leadership in global biosecurity.
So you've advised the FBI and the United Nations on these issues. What's keeping you up at night?
For me personally, it's viruses. And that's because viruses, look, there's dangerous proteins, venoms and toxins, et cetera. But those don't self-replicate. So, you'd have to have a manufacturing plant and a dispersal system. But viruses just go from host to host. And we know that a flu or a cold can spread globally in just a few weeks. So viruses right now for me are the biggest threat and I was much more optimistic pre-COVID. I was like, viruses have a lot of really beneficial characteristics as well that we use for vaccines and gene therapies. And so I was very pro-virus. I still am. But clearly through COVID and post-COVID, the threats are much better understood. And the lack of tools for monitoring and countering viral infections has been demonstrated. So we really need to work on that.
They may be the biggest risk, but we can mitigate that risk.
I think so. And again, at every level of the architecture, you look for people who are malicious. You build in constraints in the design tools that flag for anomalous or malicious behavior. You definitely have the checkpoints at synthesis and assembly. You have checkpoints in the testing and refining processes in the automated labs. And then you still need sensors everywhere in the world, which we should have, in the same way that we track weather and we track the launches of nuclear missiles. We should have biosensors that are cheap and everywhere. That's something I'd really like to see. And for me, that's just I call it the universal swab. Like right now, people get sick, and it happens all the time, whether it's a cold or a flu or RSV. And today, there's no universal swab, but a sequencer, a cheap automated DNA sequencer, say the size of a phone, would be amazing. You just swab, stick it in, and it'll tell you every virus or microbe on that swab. That to me is a technology that should exist now. It'll be in every medicine cabinet beside our thermometer, So that would be really cool. And I think we can get there if we just put a little effort into it.
So before we go to the fun questions, your team at Autodesk synthesized a virus, and did I understand the Museum of Modern Art acquired it as the first engineered organism in their collection? How did a synthetic virus end up in an art museum? Is it art or science or both? What and why?
Yeah. So at Autodesk makes computer-aided design tools. At the time, when I was working with them, they were also very interested in 3D printing. And I said, well, that's great. But, a cell is kind of the 3D printer that today's 3D printers will eventually morph into. They're multi-material. They can make more 3D printers. Anyway, so we were playing at that intersection. And I said, look, I want to 3D print biology. So can we do it? We had design software to build a virus. What we needed was the compiler. And it turned out around that time, this was 2014, some of the DNA synthesis companies said that they could start making viral sized genomes complete. Like just send them the order, they'd send me back the genome as a special project. It was still more than what they were typically offering to researchers. Short version is we took a genome that had already been synthesized in 2003 by Craig Venter. It's a viral genome that only infects bacteria. It's absolutely safe for humans. You can think of it as almost an antibiotic. It had already been made synthetically, but the Craig Venters group, he's an eminent genetic scientist. His partner was Ham Smith, who just died late last year. Ham already had a Nobel Prize. It was Ham Smith and Craig and the rest of their team at the J. Craig Venter Institute that synthesized this genome back in 2003. And they said it took them 2 weeks to do it. They didn't put any costs. They published the paper. So I said, let's do that here at Autodesk. So we got a researcher that had studied that virus. They downloaded the virus genome from GenBank, the public database of genomes. He corrected the errors in the genome in that public file. And then we sent it to all these DNA synthesis companies and said, just make it. You receive back little tubes of DNA code. And then we put that into E. coli bacteria. And we showed that, yes, some of these genomes were viable. And we took pictures of what they call the plaque assay. It's a lawn on a petri dish. It's a growth plate. It's a lawn of E. coli that gets dead spots wherever the virus booted up and killed the cells. So it's kind of like if you over-fertilize your lawn in places, it kills off the grass. So it's kind of like that. It's called a plaque assay. So we took those photos. It hit the news, which was kind of hilarious. Anyway, the curator at the Museum of Modern Art reached out to us and said, we'd love to have this organism as part of our collection. So we packaged everything up. We didn't send the virus. We sent the code. We sent the pictures. We sent the process. We sent the story, and it became part of the collection. So as far as I know, it's the first synthetic organism ever in the Museum of Modern Art in New York.
Wow. Wow. It's like, a new art movement.
It won't be the last.
It's the new cubism, right? Anyway, we always like to end with a few fun questions. What a great conversation. We could go obviously on for hours, but there'll be more to come. This has nothing to do, well, maybe it does, with all the things you've been talking about. What's the best vacation you've ever taken?
Oh my God. The best vacation. That no one has ever asked me that, by the way. I've never been a big vacation guy because my work and my life aren't separated. I keep authors hours or podcaster hours.
You see genomes everywhere. Yeah, I bet.
I do. Every time I look at something living, I just look at it as like code.
Oh my God, it must drive you crazy.
No, not really. It's just the headspace I live in. But my favorite vacations are beach vacations. I love going and playing in water. I love boogie boarding. I love just laying in the sun and reading a book. Those are the best. It is true rest. I love being by the ocean. I love the sound of waves. I could do that pretty much full time.
So, what album is the soundtrack of your life if there was one album? And you can't say Genesis.
Well, I actually have to say it's Peter Gabriel's Secret World, Secret World Live. I saw that concert live. I love that album. I've probably listened to it 1,000 times. There's something that's rewired in my brain that if I play that album, I will fall asleep. I've heard it so many times. I know every note. It just completely lulls me into relaxation and sleep.
Well, I'm going to use that instead of Ambien. That sounds way better.
Just listen to it 1,000 times. Yeah.
Yeah, I'll try that. What's something you want to get real honest about? Which, we're the Get Real podcast. Is there something you want to get real honest about with us?
Yeah. So this is what I've been working on. I am absolutely confident that the programming of life is taking off. It's off the runway. It is going. It's just going to keep going faster, better, cheaper. I love that. But what I've been thinking about now more and more is the other end of the spectrum about death. Right now, I'm thinking about how do we have more life cycles? I know that we're finite. Anything biological is finite. But I've been watching all this interest and investment in health and longevity, which is great. Like, that's such a different mindset than when I was a kid.
Totally.
I don't think we're doing it right yet. Like, this should just be baked into the health system at a very low level. We should be completely scanned and monitored periodically, by our doctors head to toe. That being said, we are going to die, but I am fascinated by the idea of backing up people today, that just a blood sample properly preserved and give you the option for a completely new life cycle through human cloning, which is not available yet, but we've been doing the same thing with animals. It's all merging together with de-extinction. Colossal, the company that has been de-extincting woolly mammoth and dire wolf and says they're going to do the dodo and others, that's cloning and editing technologies. They just acquired Biogen Pets, which is the largest pet cloning and agricultural animal cloning service in the world. Like, they do horses and other animals as well. So that now it's all become one big cloning company and editing. That is huge because now I'm starting to see the path that one day we'll do the same thing for humans. We'll become comfortable enough with this technology that just a blood sample properly stored will give you the option to having a twin brother 20, 50, 100 years from now, 1,000 years from now. You could have Jim version 1, 2, 3, 4, 5 in perpetuity.
Oh, wow. I hear you on all this. And it's fascinating and probably its own podcast. Kate, what do you think?
I'm riveted.
I know this has been, one of your areas that you're most interested in.
I also find it fascinating that what scares you most or keeps you up at night from a biosecurity standpoint is viruses. I was thinking you'd say something about warfare or chemical weapons, but really it's what changed in COVID, how we see the deadly power of viruses, which is striking to me.
Yeah. When I was younger, nuclear weapons were a big thing. That's pretty horrific. And of course, if it actually escalated into global thermonuclear war, that would be pretty bad. But those are superpowers. We have no control over those things except for who we vote for in some ways. But a virus engineered with the right characteristics, the long enough latency where it didn't show any, where it's asymptomatic, with the right effects, disability or death, could spread globally in just a few weeks and affect billions of people. And you wouldn't even know it until it kind of happened, because we don't have the tools and technologies to track just the viruses circulating around us today, even in public spaces. So that to me is the big risk factor today, potentially as many casualties as nuclear war, but with technologies that are available today by small groups. That to me is the big risk factor. Personally, I just worry I've made it now to my 60s without major problems, but I know that we're going to see more decline and eventual death. So I would love to see more and more effort go into health earlier in life.
Yeah, it's changing. Look, that's what this whole podcast is about. Early, often, prevent the event. That's the key. So thanks. I hope this inspires some of my audience to go faster and do better. And I really appreciate you spending this hour with us. Thank you.
Yeah, thank you. Really appreciate it.
Hey everyone, thanks for joining me on the Get Real podcast. I hope you feel a little healthier hanging out with me. It's going to be a lot of fun passing this along to others. Please subscribe below. Please share. I'm excited not just to talk about healthcare, but a lot of other fun topics. It's about making healthy talk about healthcare. It's not a lecture. This is supposed to be fun, and look forward to spending more time with you in the future.